Hello, and welcome to the Altasciences webinar, Conformance, From Guides to Rules, a 2020 SEND Progress Update presented by Anthony Fata. My name is Matthew Newcoop, and I am the Senior Manager of Safety Assessment at Altasciences, and I will be your moderator for this session. We have just a few announcements before we begin. The slides will advance automatically throughout the presentation. At the bottom of your browser, you will find a help icon for technical assistance. If your screen freezes or the slides do not appear to be advancing as they should, please try exiting and starting the session, as it may be an issue with your connectivity. At the end of the presentation, we will review any questions that we receive. If you have any additional questions that we are unable to answer, please feel free to contact our SEND team using the email address provided at the conclusion of the webinar. This presentation will be recorded. Before we start, a few words about Altasciences. Altasciences is a forward-thinking, mid-sized contract research organization offering pharmaceutical and biotechnology companies a proven, flexible approach to preclinical and clinical pharmacology studies, including formulation, manufacturing, and analytical services. For over 25 years, Altasciences has been partnering with sponsors to help support educated, faster, and more complete early drug development decisions. With facilities in the USA and Canada, Altasciences' full-service solutions include preclinical safety testing, clinical pharmacology and proof of concept, bioanalysis, program management, medical writing, biostatistics, manufacturing, and data management, all of which are customizable to specific sponsor requirements. Altasciences helps sponsors get better drugs to the people who need them faster. Our presenter today is Anthony Fata, Altasciences SEND Supervisor. Anthony joined Altasciences in 2017 and has over 6 years of preclinical industry experience. As Altasciences SEND expert, Anthony leads a team of specialists and plays a key role in ensuring quality and efficient SEND operations. He also acts as a SEND consultant for both internal and external stakeholders. Anthony is the member of the Clinical Data Interchange Standards Consortium SEND team. He is currently a co-lead on the Immunogenicity Specimen Assessments domain team and has co-led the Codex for SEND-ING version 3.1 in the past.
Hello all, and thank you, Matt, for the wonderful introduction. What will we be covering today? How will SEND version 3.1 impact your IND, BLA, and NDA submissions? Unpacking the biannually released FDA feedback release in the Technical Conformance Guide, also known as the TCG. What are the newly released CDISC Conformance Rules, and how are they different from what already exists? And what is the expected use of the Simplified ts.XPT with some use cases? First off, I'd like to start with a short update on SEND in 2020, specifically how SEND version 3.1 will affect your submissions. This slide shows some important dates for SEND version 3.1. Studies with their protocol signed on or after March 15, 2019 for NDA, ANDA, and BLAs, and March 15, 2020 for INDs are required to be submitted in SEND version 3.1. Expanding the scope of SEND. SEND version 3.1 expands upon the scope of SEND version 3.0 by also including GLP and non-GLP studies that are submitted into the 4.2.1.3 ECTD section for safety pharmacology. It is important to note respiratory and cardiovascular endpoints are the only ones that are currently modeled. It is also required for anything being submitted into single and repeat dose toxicity and carcinogenicity modules based on the dates from the previous slides. See the specific ECTD sections listed on the current slide. 3.0 versus 3.1, the differences. Although it added a whole new study type, SEND version 3.1 and 3.0 are similar. This slide outlines some of the example differences. There are two new domains added. The cardiovascular domain, also known as the CV domain. This domain contains cardiovascular findings. Example data that would go into this domain include non-ECG derived heart rate, systolic, and diastolic blood pressure. Additionally, there is a respiratory domain, or the RE domain. This domain contains respiratory-related findings. Example data that would go into this domain include respiratory rate and tidal volume. Most of the common tests that would go into the vital signs domain now go into either CV or RE. Body temperature is still a test that goes into the VS domain. Aside from the new domains, some minor differences include the removal of the visit day variable. But the addition of the nom-dy variable sees the intended use case of the visit day better defined in SEND version 3.1. Additionally, SEND version 3.1 allows the addition of custom domains compared to version 3.0, which is limited to only domains specified in the SENDIG. CDISC releases a short but handy guide called the Confirmed Data Endpoints, or Codex, for SEND version 3.1. This document is a great resource because it outlines types of data along with specific sections of the SENDIG that contain examples and any pertinent comments. It contains additional information regarding which tests move to which domain and what control terminology list to find them in for the CV and RE domains. In the screenshot above, you can find the test name in the far left column and a comparison of the domain and code list those tests are found in version 3.0 and version 3.1. Now I want to go back and highlight some frequently asked questions. Is SEND required for GLP and non-GLP studies? Yes, depending on if and where you're submitting that study. Would a study require SEND if it was considered GLP? If so, your non-GLP study should also be in SEND format. When will reproductive toxicology or repro/DART studies be required? As of now, repro/DART studies are not required to be submitted in SEND format. An implementation guide was released by CDISC on December 11, 2017. CDER will notify its support of the standard by placing it within the Data Standards Catalog. Will the scope of SEND continue to increase? Yes, this is just the tip of the iceberg. It will continue by expanding the number of implementation guides, including standards for dermal and ocular, as well as gene toxicity studies. Additionally, with future updates to the SENDIG, it will include more endpoints such as anti-drug antibody data. Where can I find information without having to read the SENDIGs? The Fuse Wiki FAQ is a fantastic resource. Not only does it outline frequently asked questions, but it also allows for new questions to be asked in the SEND implementation forum. Everyone should have this site bookmarked. You can find a link to it in the references section. Technical Conformance Guide and the SEND Implementation Guide Conformance Rules. As most of you know, it is almost impossible to discuss SEND in a sentence without using an acronym. From IG to TCG, the supporting documents surrounding SEND are no different. I hope to explain to you without using too many acronyms what these documents are and why they are important. Quick comparison. Here is a very brief comparison of the Technical Conformance Guide, also known as the TCG, and the SENDIG version 3.0 Conformance Rules. The TCG is released biannually by the FDA. It is a non-binding guidance used to communicate FDA preferences, and it combines SEND with SCTM and ATOM recommendations. The SENDIG Conformance Rules are released by CDISC, are rules that are based off the SEND implementation guide, and currently only have a rule set for SEND version 3.0. Technical Conformance Guide. The TCG is a vehicle for the FDA to communicate their recommendations to industry quickly. Since no standard is changed, there does not have to be a public review period. but this also means that they are non-binding and cannot cause a rejection. Note that this document contains information for SEND, SCTM, and ATOM, so it's not just for non-clinical. Here, I want to take some time to go over some of the many points discussed in the TCG. This is by no means an exhaustive list. It is not necessary to include the body weight gain domain. I believe this domain didn't provide any useful information for interpretation that could not have been recreated using the body weights domain. The comments domain, or the CO domain, should only include data relevant for study interpretation. This statement is not elaborated on, but should be a conversation between the sponsor and the CRO on what they feel comfortable including with their SEND dataset. The TCG also contains a list of parameters to be included in the trial summary domain, or TS domain, expanding upon what is listed in the implementation guide. 3 of the listed trial summary parameters in the TCG should be talked about in more detail. The pharmacological class parameter is used to describe mechanism of action, physiological effect, and/or chemical structure. The TCG guides the user to use the Veterans Administration's medication reference terminology to identify pharmacological class for established modalities. This makes finding some IND-enabling studies classes more difficult since they are, of course, not on the list. This parameter is very important when it comes to the creation of FDA, your internal, and/or third-party SEND databases or warehouses. Pharmacological class would be useful to search through a library of datasets/compounds to pull similar studies in order to compare possible toxicological effects across a class. It is very important to discuss with your CRO or SEND dataset provider what you want them to input into this field to ensure the most accurate descriptor is used, creating the best possible indexability for future databases. Primary treatment unique ingredient IDs are a unique ISO identifier for compounds maintained jointly by the FDA, NIH, and EMA. Please provide these to your CRO or SEND dataset provider, as it's always best to include them if they are known. Additionally, primary treatment cache registry number is assigned to chemical compounds by the American Chemical Society. Also, please provide these to your CRO or SEND dataset provider if known. For measurements that occur over multiple study days but are grouped for analysis, it should be populated with only a single day within the visit day or non-DY variables for SEND version 3.0 versus 3.1 respectively. An example is a blood collection for a drug plasma concentration that occurs at 2, 12, and 24 hours after the first dose. The visit day, or non-DY, should be 1 for all 3 time points, even though the study day, DY, for the 24-hour time point will be 2. Specifically, within the laboratory test results, or LB domain, any categorical numeric values should not have LB stress n filled in. Even if the value contains numbers, they are not necessarily numeric. This will cause a warning when using some validators. An example of this would be urine pH based on a dipstick. You will see an example of this on the next slide. Here is an example of the LB domain with sub-urinalysis values. You can see they are numeric, but since they are categorical, they do not have any values within the LB stress n column, highlighted by the arrow on the slide. The pharmacokinetic concentration, or PC domain, has had a couple requested changes from what is specified within the IG. When a drug concentration within the PC domain is below the limit of quantification, the PC ORAs should remain exactly what is reported from the analyzer, while the PC stress C should be BLQ, which stands for below the limit of quantification. If the BLQ value is used for analysis within the TK profile and assigned a number, then that assigned number should be populated in a SUP-PC domain. But if the BLQ value is excluded completely from the TK profile, then no SUP-PC is required. In this example of a PC domain, all of the animals were below the limit of quantification. You can see that the PC ORES remained the same as what was outputted from the analyzer, while the PC stress C, which was highlighted in the arrow at the bottom, was changed to BLQ. Additionally, while the visit day or non-DY remains 1 for this whole time series up to the last 168-hour time point. Continuing the previous example, this SUP-PC domain shows the numeric values of BLQ that are being fed into the non-compartmental TK analysis. In this case, it is the lower limit of quantification of the test, but it could be any value that is used in the non-compartmental analysis. Now, switching gears to the SEN version 3.0 conformance rules. The rules were finalized/published in March 2020 by CDISC. A version for SEN version 3.1 is currently in development. The creation process was a line-by-line detailing of every statement within the SEND implementation guide to see if it qualified as an implementable rule. An example of this is that test CDs cannot be longer than 8 characters. The conformance rules are not to be confused with current validator or business rules. In the future, it is possible that the rules are to be implemented into the FDA validator rules. Here is an example of the conformance rules released by CDISC showing 2 rules related to test CD across all domains. The first one states that test CD cannot be longer than 8 characters. The second one states that test CD cannot start with a number. Other rules. Of course, there is not only one set of rules. In addition to the conformance rules, other rules exist, including the FDA business rules, as well as other validator rules. FDA business rules are simplified general requirements written for the FDA to internally use and process SEND data submissions. Validator rules are machine readable and used by software to run against datasets. An example being the Pinnacle 21 validator rules. Now, I would like to move on and discuss the use of simplified ts.XPT files. The eCTD system automatically checks submissions for trial summary or ts.xpt files when any of the following file tags are used: preclinical study report, legacy clinical study report body. Within the following modules for single and repeat dose toxicity and carcinogenicity. There are multiple instances where a report is submitted without requiring a SEND dataset. In those cases, one of 2 types of simplified ts.xpts should be included. Simplified ts.xpt with a date. Here is an example of a simplified ts.xpt where the study type is modeled in SEND, but the study does not have to be submitted in SEND format. The TS val is filled with the protocol signature date, while the TS val NF is left blank. Here is an example of a simplified ts.xpt with a null value. Note that the TS val is left blank, but the TS val NF is filled in with an NA or a not applicable. Used in a variety of cases where study initiation is not applicable, some examples include, but are not limited to, non-clinical safety reports, carcinogenicity protocol amendments or carcinogenicity risk assessments, exploratory or tolerability toxicity study summaries, literature study reports specified used as non-clinical support for safety, specialized toxicity studies conducted where there are no study parameters modeled in the applicable SENDIG. Here is a flowchart with some example cases of using a simplified ts.xpt domain. When submitting into the following modules and the file tag of Legacy Clinical Study Report is used for a literature study, a simplified ts.xpt with null value should be submitted. When a file tag of Preclinical Study Report is used for a repeat dose toxicity study that had its protocol signed before December 17, 2017, a simplified ts.xpt with a date can be submitted since this study is model for SEND but was before the requirement date. When a file tag of study report body is used for an expert pathologist report, a simplified ts.xpt with null value would be submitted. Note, these are all examples. We have reached the end of the new content of the webinar. I hope that you all have learned a little bit more about SEND during this time. Specifically, how safety pharmacology studies are now included with SEND version 3.1, a summary of some of the SEND-specific content within the TCG and how that differs from the CDISC Conformance Rules, and how simplified ts.xpts should be used within the ECTD to avoid a technical rejection. Here are the helpful references that I used to create this webinar today. The first link takes you to the Technical Conformance Guide. It was recently updated in March 2020. Below is a main CDISC link, as well as links to the individual conformance rules and IG for 3.0 and 3.1. Additionally, there is the specification for the ECTD and study tagging files for the FDA, the FDA technical rejection criteria, as well as the CDER and CBER guide to electronic data submission within the ECTD. Last but not least, the Fuse Wiki FAQ. This link is very helpful for anyone who's just starting off with SEND or has questions that they think that others might have asked.
That brings us to the end of the webinar. Now we would like to go back and answer some questions that were submitted regarding this topic. First question for you, Tony. Do I have to go back to older studies and have them redone in SEND 3.1?
No, absolutely not, Matt. This would be a humongous undertaking for industry, and I think that I might voluntarily enter early retirement. For SEND, studies are considered only based on their protocol signature date. This means you do not have to go back and update any studies from version 3.0 to version 3.1. You can submit with an application with multiple versions of SEND.
Excellent, thank you. Next question, who determines what data should be included in the CO domain that you discussed earlier?
This should be a conversation between the CRO and the sponsor to see what they feel comfortable with including. As of now, the SEND team is making a determination, but I'm very much open and willing to involve the sponsor to see what they want included or not.
So, Tony, is the Technical Conformance Guide basically a precursor to the CDISC rules? Should everything just adhere to the guide as well as the rules?
No. Since the CDISC compliance rules are completely based off the IG, there should be no impact of the TCG on the rules. But since the TCG are preferences of the FDA, I can see them having impact on future versions of the SEND IGs.
All right, thank you. So next question, Tony, how can I produce the simplified ts.xpt domains?
Well, there's lots of options. You can produce them in-house. You can ask your preferred CRO, including us, Altasciences, or you can ask a third-party conversion company. There are lots of companies out there who will do it for you, and these are pretty easy files to create since, as you saw before, they're just one line. So please reach out to any and all your SEND providers, and they'd be happy to help you with that.
Okay, thank you. All right, next question, Tony. Other than submitting the studies, what else could I be doing with them?
So data is power, and I think that data warehousing will pay dividends in the future. Using Send datasets for visualization is a really powerful tool, not only to gain insight into individual studies, but some visualization tools have the ability to visualize across multiple studies, and this allows you to see the effect across multiple studies or multiple compounds. Additionally, why not utilize these packages that you're already paying for and creating for a submission?
So just to clarify, Send is not just for submissions, is that correct?
Absolutely not. You should be using it in-house. Whether you see the benefit today is a question, but I think in the future there will definitely be a benefit.
Awesome, thank you. And last question I have here, will Send be required to other countries or regulatory bodies other than the FDA?
So both the Japanese Pharmaceutical and Medical Device Agency, or the PMDA, and the European Medicines Agency, or the EMA, have not formally required Send, but I have the feeling that they're looking at the US to kind of iron out some kinks before they formally adopt it themselves. And another side fact is that CBER, another office within the FDA, is also currently considering adopting Send to fit their needs.
Excellent, thank you. This concludes our webinar, Conformance from Guides to Rules, a 2020 Send Progress Update, presented by Anthony Fata. The slides that were presented will be made available for attendees for your reference, and they will be made available on the Altasciences website. I would personally like to thank Anthony for taking the time to present this information to us. If you have any questions regarding the content of this webinar or Send in general, please reach out to Anthony using the email provided on your screen. On behalf of Altasciences, I would like to thank you all for attending and have a great day.